In this lab session, you will be performing an endospore stain. The endospore stain is a differential stain that may be performed to detect the presence and location of endospores in bacterial cells and to differentiate between endospore-forming bacteria and non-endospore-forming bacteria. Endospores are protective structures formed by a few genera of bacteria. Endospores differ from vegetative bacterial cells. Vegetative bacterial cells are cells that are metabolically active and therefore require a favorable environment and adequate nutrition. Endospores are produced from vegetative cells through a process known as sporulation, which is initiated when the vegetative cells encounter harsh conditions, such as a lack of nutrients. During sporulation, the vegetative cell is referred to as a sporangium, which forms the endospore within itself. After the endospore is fully formed, the sporangium is sloughed off. The endospore is a metabolically inactive structure made of several layers of resistant coats that surround and protect the DNA, some ribosomes, RNA, and enzymes. The outer coating of the endospore is a tough coat composed of a number of proteins, including a keratin-like protein, which is resistant to heat, radiation, chemical disinfectants, and desiccation. When the endospore is exposed to favorable conditions again, it germinates to produce a vegetative cell. Endospores are sometimes referred to as spores. It is important not to confuse bacterial endospores with fungal spores. Fungi use spores as a means of reproduction. Bacterial endospores strictly serve a protective function and play absolutely no role in reproduction. They simply serve as a means of survival in harsh conditions. Bacterial endospores are produced by only a few genera of bacteria. Two that you will become familiar with are the genus Bacillus and the genus Clostridium. Most members of the genus Bacillus are found living as soil, freshwater, or marine saprophytes. Saprophytes are organisms that feed on dead decaying material, therefore acting as decomposers. One notable human pathogen in this genus is Bacillus anthracis, the causative agent of anthrax. Most members of the genus Clostridium are soil or aquatic saprophytes, while others inhabit the human intestines. Some notable pathogens are Clostridium tetani, Clostridium botulinum, Clostridium perfringens, and Clostridium difficile, which cause tetanus, botulism, gas gangrene, and pseudomembranous colitis, respectively. Since few medically important bacteria are capable of forming endospores, determining that an unknown sample has the ability to produce endospores aids greatly in the identification process. To determine whether or not a bacteria in an unknown sample produces endospores, we would perform the endospore stain. This is because endospores are very resistant to staining. In fact, a gram stain of an endospore-forming bacteria can hint at the presence of endospores. In the staining process, the occasional formation of endospores would be observed as clear areas within the cells, because the crystal violet iodine complex does not penetrate the protective coats of the endospores. Such a result would then suggest the need to perform an endospore stain. The endospore stain method outlined in your NCTC lab manual is the Schaefer-Fulton method. In this method, the primary stain, malachite green, is applied to a heat-fixed bacterial sample and then exposed to steam heat to drive the malachite green into the protective coating of the endospore. Malachite green is an aqueous stain that does not bind strongly to cellular structures. Its affinity for cellular structures is very low. So as water is applied as a decolorizer, malachite green is easily washed out of the vegetative cells, leaving them clear, while the protective coat of the endospore traps the malachite green, leaving the endospore green. The counterstained safranin is then applied to the smear. Safranin cannot penetrate the endospore, but instead serves to stain vegetative cells. So at the end of the staining process, vegetative cells and sporangium appear pink and endospores appear green. The results of the endospore stain allow us to determine whether or not the bacterial sample is capable of producing endospores. In addition, if endospores are present, it also allows us to gain information regarding their size, shape, and relative location within the sporangium, which are important details that help us in the identification of the species. Three terms are utilized to describe the location of endospores within sporangia. Centrally located endospores form in the middle of the sporangium. Terminal endospores form at the extreme end of the sporangium, and subterminal form between the end and the middle of the cell. The endospores will be spherical or elliptical in shape, and upon observation you will see that small endospores do not affect the shape of the sporangium, while large endospores cause bulging of the sporangium. In your lab session, you will prepare 3 bacterial smears of Bacillus subtilis. Your 3 samples will vary in their age. You will have a young sample, an old sample, and a very old sample. The old and very old samples have been left in the same media for an extended period of time, so it would be expected that the bacteria in those cultures would have exhausted most of their nutrients. The lack of nutrients, if you recall, is a trigger for sporulation. Based on this information, you should see more endospores present in samples as they age.